So the title of my talk today is Coal-Map-2, Mapping the Genomic Architecture of Adaptive Traits with Interspecific and Progressive Origin. So before I go into the details of my method, I'm going to define what is introgression first. Introgression involves the transfer of genetic information between two species as a result of hybridization and repeated backcrossing. If you take a look at the following example, we have two species, species A and species B. If we cross those two species together, we get a first generation hybrid where almost half of the genome comes from species A and the other half comes from species B. Now, if you repeatedly backcross this hybrid with species A, what you end up with is an introgressed hybrid where more than half of its genome comes from species A, and there are some introgressed regions coming from species B. Now, adaptive interspecific introgression has played a key role in the evolution of many novel traits in different eukaryotic organisms. For example, there are studies that show or that suggest that there is an increase in hemoglobin concentration in Tibetans as an adaptation to high altitude environments. This is the result of introgression of some DNA segments from Denisovan individuals into humans. Another example of adaptive interspecific introgression involves its role in the evolution of mimetic butterfly wing patterns. Furthermore, interspecific introgression has played a key role in the evolution of pesticide-resistant house mice, where basically mice share genes that confer for warfarin resistance in order to survive. If you take a look at the following example, we have a house mouse that actually lacks the warfarin resistance genes. We have another mouse species that carries the warfarin resistance genes. If you cross those two mouse species together, you get a hybrid species that actually carries the warfarin resistant genes. So basically, this example, as well as the aforementioned two examples, are a subset of many cases, many examples where interspecific introgression or adaptive interspecific introgression has played a key role in the evolution of novel traits. Now, going to the motivation of my work, I'm interested in identifying the genomic architecture of those introgressed phenotypic traits. So basically, to address this question, there are many methods that have been developed. These methods differ in terms of the research time, the resolution, as well as the allele number, or the number of alleles that could be sampled per locus. If you take a look at the following figure, the y-axis shows the research time measured in years. The x-axis shows the resolution measured in base pairs, and then you have different methods. Each method is colored according to the number of alleles that could be sampled per locus. So basically, based on this figure, we can observe that association mapping offers the highest resolution, the shortest amount of research time, as well as the highest allele number. So to address this fundamental question, we basically use association mapping, or we focus on association mapping. What is association mapping? So the goal of association mapping is basically to identify the underlying genetic factors contributing to a variation in phenotype of interest. An input to an association mapping experiment is usually a multiple sequence alignment, that's your genotype, as well as a phenotypic vector, that's your trait, could be quantitative or qualitative. On the other hand, the output are a set of sites, you can think of as a set of SNPs that are associated with the phenotype or with the trait. Now, a typical association mapping experiment is usually conducted as follows. We first obtain and phenotype the population of samples. We then obtain the genotype, which is a multiple sequence alignment in this case. The third step is to conduct association mapping or apply the association mapping method on the data, basically to identify any possible statistical associations between genotypic and phenotypic characters. And finally, the last step is to examine the implicated regions and identify any possible candidate genes that are associated with the trait. Now, one of the key issues that need to be addressed when conducting any association mapping experiment is sample relatedness. Sample relatedness or population structure could induce spurious associations between genotypic and phenotypic characters when the actual evolutionary relatedness is not accounted for or is modeled incorrectly. If you take a look at the following figure, we have a tree. We have a set of samples. Each sample is colored according to its phenotype, which happens to be body weight in this case. Basically, you can see that samples located in the smaller plate are more closely related to one another than samples located in the larger plate. So we do have population structure, we do have sample relatedness. Now, if you apply an association mapping method without modeling or without correctly modeling this sample relatedness that you see in this figure, then you end up with the following Manhattan plot, where it seems that every locus seems to be associated with the phenotype or body weight. So there's a lot of spurious associations, a lot of false positives. On the other hand, if you actually apply an association mapping method while correctly modeling the evolutionary relatedness that you see in this figure, then you end up with this second Manhattan plot, where it seems that there is only one locus that is associated with the phenotype. So it is very important to model sample relatedness correctly in order to generate reasonable as well as reliable results. Now, there are many methods that have been developed which model evolutionary relatedness between samples. I group these methods into two groups. There are methods that infer and model structure. Most notably, there is a method called Eigenstrat, which applies principal component analysis to derive the top principal components and then use these top principal components to correct for population structure. And then there is another set of methods that actually utilize a kinship matrix. A kinship matrix is simply a matrix that describes the genetic similarity between individuals in your population. Now, the common thing about all these state-of-the-art methods is that they all assume that the evolutionary history of all genomic loci is described by a single phylogenetic tree. So they use a single phylogenetic tree to describe the relatedness between the sampled individuals. What I'll show in the next slide is that this assumption does not always hold. This is a figure showing genomic regions in the mouse species known as Mus musculus that were inferred to originate via introgression from another mouse species known as Mus pretus. The y-axis shows the different mice labeled according to their country of origin, and then the x-axis shows the genomic positions. The colored regions in the figure show the introgressed regions. On the other hand, the non-colored or the white regions show the non-introgressed regions. A couple of key points from this figure. The sample relatedness or the structure in the introgressed region differs drastically from the sample relatedness in the non-introgressed region. Additionally, the sample relatedness in the introgressed as well as the non-introgressed regions differs drastically from the global sample relatedness. Basically, the global sample relatedness is the sample relatedness measured across all loci, all sites. So, to model the sample relatedness correctly, you need to model the sample relatedness in the introgressed region, in the non-introgressed regions, as well as account for the global sample relatedness. This is where our method comes into play. So I'm going to talk a bit about the motivation of our newly introduced method. As I said, state-of-the-art methods assume that the evolutionary history of all genomic loci are described by a single phylogenetic tree. In other words, they only account for global sample relatedness, which is the sample relatedness measured across all loci. What we need is a method that accounts for both global as well as local sample relatedness. It should account for the introgressed as well as the non-introgressed regions. This is where Coal-Map-2 comes into place. So basically, we developed Coal-Map-2, which captures both global as well as local sample relatedness. Basically, Coal-Map-2 utilizes a linear mixed model to evaluate the relationship between the phenotype Y and the test locus xj. In this equation, W is a fixed effect matrix containing covariates, which basically are used to model the local sample relatedness or the sample relatedness in the introgressed regions. Alpha are the coefficients of these covariates located in W. Beta is the effect size of test locus xj. Now U is a random effect modeled as a multivariate normal distribution. Basically, U is used to model the global sample relatedness. And finally, we have epsilon, which is also a random effect modeled as a multivariate normal distribution, and it is used to model any unexplained variation in the phenotype Y. Now, it is important to note one thing here. This linear mixed model should only, or this W, this covariate matrix W, should only contribute to the linear mixed model when the current scanned locus contains causal SNPs. So what we did is we conducted a simulation study. We simulated a multiple sequence alignment. We tried to use simulation conditions that are based on some empirical studies of introgression in mice. We used the trait model, which is an extension of the trait model used in Longano 1999. Basically, this trait model has two components, a genotypic component as well as an environmental component. The genotypic component is basically weighted by pi, which is simply the trait or the proportion of trait variation contributed by the genotypic effect. We used two different trait model conditions. They basically selected causal sites from one locus or two loci. So the following figure shows the results after applying Coal-Map-2 on this simulated data set that we tested using hybridization frequency value gamma of 0.5. We have two figures here. The first figure corresponds to the single causal locus trait model condition. The second figure corresponds to the two causal loci trait model condition. Now, the y-axis shows the true positive rate, the x-axis shows the false positive rate. These are simply receiver operating characteristic curves. The magenta line or the purple line shows the performance of Coal-Map-2. The blue line shows, or the blue curve shows the performance of Coal-Map, which is the predecessor method of Coal-Map-2. And finally, the red curve shows the performance of Eigenstrat. We compared our method to Eigenstrat, given that Eigenstrat is one of the popular methods, given its statistical power as well as the false positive control that it has. So what we did is the following. We compared the performance advantage of our method, Coal-Map-2, compared to Coal-Map and Eigenstrat using the long and old test. And we find that our Coal-Map-2 significantly outperforms both Coal-Map as well as Eigenstrat, using both the single causal locus trait model condition or the two causal loci trait model condition. So in conclusion, we know that current state-of-the-art methods do not account for local genealogical variation. They basically account for sample relatedness, but they only account for the global sample relatedness, which is the sample relatedness measured across all loci. What we do in this study is we develop a method, a linear mixed model, that actually accounts for both the global as well as the local sample relatedness. This is why we introduced Coal-Map-2, which is a new association mapping method. We showed that the performance of Coal-Map-2 is comparable or better than Coal-Map or Eigenstrat, using or in terms of statistical power as well as false positive rate. Finally, in this talk, I mainly focused on introgression. However, I'd like to make the point that Coal-Map-2 can be generalized and applied on other traits with different complex evolutionary origins. So it doesn't have to be introgression. Now, you can find the project webpage as well as the software on the following GitLab page. And if you have any questions, I'd be more than happy to answer them. Thank you very much.